Hello, good morning, everyone. I hope you are all doing. You're most welcome to the workshop of Enzyme Engineering. In this, our second batch in June, we had also these small workshops before in the May batch. So it's a small group. 13 students are there. So far, 10 students have joined us. So everyone, you're most welcome here. So there were some questions. I think you are not aware of the how the sessions are going, how things will be going. So I had some questions being asked in the group. So most likely we will start by 7 or 7:05, and then we will wait for 5 to 10 minutes in order to let all join in the starting initial period, right? So and then we initiate our lectures, and the platform will be only YouTube. There are three platforms that I discuss in my all lectures. That Yeah, first of all, are you able to see my screen, and are you able to listen to my voice clearly? These two things are the basic things for our online classes nowadays. So if these two things are good, give me a solid thumbs up. If my voice and screen is clear, give me a solid thumbs up, and then we could initiate and discuss further how the workshop will go. Good, good. Everybody, please give me it. And the language, the communication, should we keep it in English or mix Hindi? I'm comfortable in both. We keep it in English or English, Hindi English mix. Okay, okay. Yeah, most of my sessions they are in English only. That's why it's okay. So it will be in English. And second thing is, um, the there are many platforms to have online classes like Google what is it called? Google Meet, Zoom sessions, right? Many others. The problem with all other sessions are that the recording, it's being done in breakage. So in Zoom you can have recording for 40 minutes only, then the sessions gets over, right? And then it doesn't make sense to stop the class, then start again, then stop the class, then start again. So because we're gonna have this 2 hours class, and we also, whenever someone wants to get in by mistake because of network issue or something, that we get to log out from the sessions during these. And then if we are not present on the screen, we didn't let the student add into that. So there are many problems with them. So overall, with my own experience from 1 year class, I have chosen this YouTube as a best platform so far. We can have continuous streaming these lectures. So I have made this into the playlist called Enzyme Engineering and Technology June batch, this workshop for the June batch. So each date will be there correspondingly, and you can come and watch it again. Cool. So that was voice is fine, screen is fine, language is English, streaming medium would be the YouTube. Okay, all these things runs. Now we will have our just first 15 to 20 minutes just to get to know about each other, who we are, and yeah, from where we belong to, what our hobbies are, what we have done in our life, just to get to know each other, so that we can have a kick start to our workshop. So basically, I had my BTech in biotechnology from Punjab Technical University in Punjab. Then after finishing my degree, I worked for as a lecturer for 5 months at Singh College of Engineering and Technology for 5 months. And then during that period, I got a chance to apply for my masters. During these, after the BTech, within the 6 months, I applied for my MSc Agrobiotechnology at Justus Liebig University, Gießen, Germany. So I went there to Germany for like 2010 till 2012 for my masters. But the transgenic crops were not so hip into the Euro people, so they don't like to eat GMOs and all those. So when we tried to put them for the into the trial for the fields, trial into the field, but everyone, all the local people, they came, they start to burn it up. So I thought that's not my future, is where people don't like to eat something I'm working my day and night. So I switched to human medicine. I got a chance to work at the Max Planck Institute of Lung and Research. It's in Bad Nauheim. So there I worked for like 1 year as a research assistant to a PhD student, Dagmar, in the histology department. And based on that, I got my admission in the uh, pharmacological unit for my master thesis in the case of cancer oncology field. Then finishing my master thesis, then I went based on that, got my PhD admission in the same year 2013, as the in the molecular medicine, where my topic was related to the redox regulation of protein phosphatase in cardiomyocytes. So the scholarship was for 3 years, but it took me around 4.5 years to complete my PhD. So after finishing everything, so living in Germany for 7 years, I came back to India. Then I joined assistant professor at Mata Gujari College, where I was teaching various subjects of biotechnology. And then also in 2020, I joined EduFabrica, so where I'm teaching at the moment. I have already brought, actually, I initiated with Vatsal sir this biological units. So I brought MBBT, biochemistry, proteomics, principle of plant biotechnology, principle of genetics, which is being well flourished going on as a 1 month course. Um, yeah, so at the moment I brought these three new more subjects, that is enzyme engineering, molecular biology, as well as medicinal biochemistry. So it's just growing up with a small baby to big uh, within the EduFabrica. And also I was part of assistant professor at Rama University in Kanpur, where I was teaching and being part of head and training placement cell, timetable in charge, and many other stuff too. So something about me. So yeah, techniques and methods, academic achievements, blah blah blah. That's something beyond. Okay, so I want to discuss now what will be the. I hope these objectives have been shared with you. Historical perspective, enzyme classification, enzyme chemistry, isolation, turnover number, marker enzyme. So I will deal in within these 2 days basically the basics of enzyme. Yeah, what the enzyme is, what's the thing are. So my main concern is always in teaching at the moment is to get to know the students the basics first, so that we could go into the advanced version later. Because sometimes our basics are not so clear, which made us to like understand the difficult part very difficult. So that's the objective it would be. So yeah, earlier we have thought to make these workshop for 2 days, but I suggested to make it to 3 days, each day 2 hours, so that we can deliver the lectures in more easy way, not so haphazard way. Earlier I was thinking to do it in a 1 day, but students, as you all suggested, it should be like we have classes and everything, so we make it to 3 days. So our classes will be from 7 a.m. till 9 a.m. every day for 3 days: Friday, Saturday, and Sunday. So I will, whatever the things that I will be talking to you here, so I will keep sharing if I have discussed anything with you. Yeah. So I will share it just over the group then. Whether it's presentations, because my way of teaching would be mainly the presentations will be there. So once the presentation is over, so I will share that PPT into the group, so that side by side you can have a good, very solid um study material also. So I have plenty of study material for all of you within these 3 days. So I have some really good books also, some good MCQs, yeah, some good amounts of MCQs, and yeah, and plenty of many things. You will see in upcoming days. Okay, so that was about objective. We have discussed about me, who am I. We have discussed about uh, yeah, what we'll be doing in upcoming 3 days. We have discussed about the timings. Okay, now next is yours. It's your turns now, students. Give me your introduction. In the introduction, let me know, because in the yeah, your name, your like course, what you're studying, your year, your place, your hobbies, and why did you choose the course, not my course, but the course that you are doing of your own, and where do you see in upcoming 5 years. So think about it, um, over these questions, and then reply to them. So everyone, each one of them, I need answers from my each students here, and then we will proceed further. Answer them, students. Tell me, students, I'm waiting for your answer. Okay. Soumya Mahapatra, um, postgraduate, first year biotech, Odisha. I have chosen not this course, bache, but the course that you are doing actually. Of your own, that's why I wrote. Pure organic chemistry. Okay, most welcome, Archana. Ah, you are a PhD third year. Oh, so soon you might getting your PhD then. Welcome to the club. Praditi Das, B Pharma plus MBA. Oh wow, that's a great combination. Hobbies, listening to music, interest in sports like volleyball and kabaddi. Oh, that's good. Oh, marketing department in Abort. That's interesting. Oh, I have seen these news. It's a very sad time going on, Paulmi Bagachi. Um, because of COVID, we have lost our near and dear ones in this time. But we have to continue our life and don't want to stop because MBBS or the allied field of life sciences, they are not so different from each other. So people who are doing this genetic engineering or any biotech related fields, they are still getting the same positions in their life. So keep thinking high, keep pricing high. Anushka Das, BSc honors, Vidyasagar University, West Bengal. Hmm. Shreya Das, Physiology, West Bengal. PhD degree and researcher. Oh, most welcome. You're all the best for that. My best wishes for you from all of us. Bhargavi Rao. B Pharma, Chennai. Pharmacology. Oh, chemistry and pharmacology. Good. hmm So mostly I can see they are we are into the research. Some are MBBS, some are in bachelor's, some are in masters. So it's a mixed group, and I can see mostly from the Bengal side. I can see here, West Bengal. That's good. You're most welcome. Cool. Um, Everyone, most welcome. So based on that, we will continue our journey. We'll start with our lectures now. So there will be some breaks also, breakout sessions. We will not continue more than 40 minutes. So that's the major psychology of a human or student says that only student could grab about anything only for 40 minutes. Then it start to lose its interest and we cannot focus. It's for everyone. Whether you're watching movie or whether you are doing anything of your interest, of your passion, you can just get in there for 40 minutes only, then we start to loosen up things. So we will have the lectures for 40 minutes to 15 minutes first, then we will take some breaks and then continue with the another lecture. Okay, so get up, get your coffee, tea, green tea, whatever is your favorite beverage in the morning, and then yeah, let's start the journey. I was looking for this thumbnail. I think I cannot change the thumbnail now. Or was my photo is coming up there? Just a second. Yeah. So that's the session. We will discuss basically about the history of enzyme engineering first. Then we will continue the background of enzyme engineering and then some fundamentals of protein chemistry. And then gradually we go for more deeper and deeper points. So basically, what are enzymes are? Basically, enzymes are the one which helps to fasten up reactions. They act as a biological catalyst. So they come into the reaction. Yeah, they come into the reaction. They took part, and they make the substrate into the product, and they get used in that, but never get destroyed. They keep up reusing and reusing them. We keep reusing them, these enzymes. So that's the basic work of enzyme. If there is enzyme in our body, only then there is life. If our body doesn't have enzyme, so our body is not there anymore. So enzymes plays a very essential role in human body, and also beyond that, from the industrial perspective, it has made many people's life very easy, believe me. Whether it's the food industry, leather industry, or any detergent industry, it's being part of each industry, and everyone are dependent upon these enzymes. Yeah. So in earlier years, so people were using them, like in the case of like Nirma powder, let's say. So no, but nobody was understanding that how, what is the mechanism behind it, how they are cleaning the clothes. So it was all later and later things got start to develop. People start to understand its importance in the human body or in other applications with time. And this doesn't come in a single day. It took quite number of years in order to understand that how the kinetics of enzyme engineering works. With the Michaelis and Menton coming up, Line Weaver, Burke, and then they coming up to produce the various reactions of how the enzymes are working. Yeah, so all these things matters a lot, and they come gradually, each step one by one, not in a single day. Let's start history of enzyme engineering. So enzymes are proteins that catalyze, that is, increase the rate of chemical reactions, as we discussed. So they are the one which come act into the reaction. They do their role and make the reaction fast, and they again start working again in that. That's basically enzymes are. And enzyme engineering, it's the application in which we understand how we can modify these enzyme structures, how we can modify the catalytic activity of these isolated enzymes. So mainly dealing with the enzyme structures and catalytic part, the active site. Enzymes have these active sites where the substrates get attached and start to produce products out of it. So they start to these with the help of enzymes. They give new metabolites. They allow to happen new reactions, and they convert compounds from one form to another. Whether it's environmental perspective, yeah. So there's called thing called bioleaching or bioremediation, biodegradations, bionitrifications. So all these reactions in environmental also, the enzyme engineering is playing a role. So there's not a single field that I can speak like their enzymes are not being used. So even in your molecular biology, if you see the DNA ligase, DNA polymerase, RNAs, yeah. So during the DNA replication, these helicase, topoisomerase, all these enzymes, they are making a good effort to do the happen this semi-conservative DNA replication in the human body. Not only that, then let's say amylase, trypsinase, and then in the digestive system, peptinase. So many enzymes are also there in order to digest our food. So in earlier years, if we see from the past, from the history of biotechnology, that before Christ, where it was being used, mostly enzymes. So they were making breads, beer using yeast in the Egypt. So in the Egypt, they were using a biotechnology to be used for bread, beer, and using yeast, and also for production of cheese, wine was done in Sumeria, China, and Egypt. Yeah, this fermentation of grapes. red wine, white wine, many more. Then initially, the first vaccination that happened in 1797 by Edward Jenner, who lived from 1749 to 1823. He was an English scientist who have discovered the first smallpox vaccine at that time, and also known to be as a father of immunology. So we can say that from the perspective of biotechnology, he was the first one who initiated these vaccinations to bring these attenuated viruses into the body and let the antibody produce in our body afterwards, and then we can fight against the disease. If let's say if this guy, our Edward Jenner, if he hasn't discovered this vaccine, and there's no immunology field was discovered, so this pandemic that we had at now in 2020 and 2021, that is COVID, we might not have discovered any uh, diagnosis for that, the vaccinated that we have discovered, and we don't know yet whether Covaxin and Covishield that has been discovered just now, whether they are working or not. Because most of the people, they're saying when they are getting vaccinated, even after 2 doses, the antibodies are not being produced in their body. It's just they're getting water maybe in their body. And the Covaxin, they are being on the clinical trials at the moment in India. So whosoever is getting it, so we are getting to know what is happening to the human being. So we are like guinea pigs at the moment of the government. That's the reality. We are at the moment, whosoever is getting the vaccine, we are like a guinea pig. If anything bad happens to us, anything, so it will be recorded in the government chapter that this person has these side effects of the vaccine. So far nothing came out to be serious. Even I got my first shot. My parents got the double shots. So let's see how things works out in upcoming days. And did you also got their vaccination? Please comment in your box. And what is your views about the vaccination? Whether we should get it or not. Somehow it could save the severe effect of the COVID on the long term. 80% there are chances that you will not get the COVID after the vaccination, but there will be chances that the vaccination itself have some side effects on our body. So there are two ways. Just in the comments, give me your views. So I will read them later and we can discuss about it. Then father of genetics, who is known to work on the peas, Hungarian scientist in Austria, father, a priest, Baptist, Gregor Johann Mendel. Yeah. So he is known for working on this extensively on the field of genetics in his backyard, um, and he brought really good results and during his time. So that's what happened with the scientists. So what happened, whatever he discovered in his time, it was not recognized, yeah, or in the scientific community. It was, they said it's all rubbish, it doesn't make sense. Law of segregation, law of dominance, yeah, all his laws were not into the people's eye. It was just waste of time for them. But later on, like three more scientists came. They worked on his work on other models, Drosophila melongster, and they discovered that what he was saying was very remarkable of his time. It was something he spoke in advance, that how genetics works. So then later on his work was republished and was got to know in the public, and it was, yeah, then he became as a father of genetics also. So that's really happened in science. When you do your research, it will be not recognized at that time, but later 10 years, 20 years, people will get to know about your research. Then comes your first alcoholic respiration with cell-free extracts. This was Edward Buchner from 1860s to 1970. He was a German chemist, the winner of 1907 Nobel Prize winner in chemistry on the work of fermentation. Then this lock and key model of enzymes by Hermann Emil Fischer from 1877 to 1947. He was also, yeah, that's, I want to admit, you can see that most of the Austria is also nearby German. Yeah, this is his English, British. These are Germans. So Germans are the one, they are very known to be good in as a researcher. Good as a, also they got many Nobel Prize winner in the past. Um, and they had a really good sense of science when they come into the research, because I have closely worked with them during my research time. And yeah, undoubtedly they are good in that field. So he's the one, Hermann Emil Fischer. He worked on the lock and key model, um, proposed a substrate and enzyme interaction, and he is the winner of also in 1902 Nobel Prize winner in the chemistry. Now this lock and key model we will discuss. You might be aware also that the your locks are like enzyme and key is like a substrate, and only the key will get into that lock only, of which its structure is made up of. It is meant to be open. Only those keys will fit into the lock. The same way substrate will attach to only those enzymes in which enzyme have the modifications of the structure according to the substrate. Only then reaction will takes place. Then in 1982, antibiotics were discovered by Sir Alexander Fleming. He's a Scottish biologist, pharmacologist, and he also got the Nobel Prize winner in 1945 in physiology of medicine. Then your this Frederick Sanger, he got actually two Nobel Prize winner, one in 1958 and one in 1980. One, he got Nobel Prize winner for determining the sequence of insulin, that how the insulin looks like, and then later into the other field of its uh, DNA technology itself. So he got twice Nobel Prize winner during his lifetime. He was working in the MRC laboratory, if you have heard of MRCs. So that was the era where lot of things were working in the field of major biotechnology, that the things were developing according to me. Then there was 1953. That's the era where the real biotechnology started. We got to know about DNA structure. Yeah, so James D. Watson and Francis Crick. So they proposed this DNA structure, and they jointly got the Nobel Prize winner in 1962 for physiology or medicine. So there is a Rosalind Franklin who first initially discovered the X-ray structure of DNA, but he couldn't understand that what is what in it, what things they are made up of, what are nucleotides, what are sugars, what are phosphates, what are hydrogen bonds, what are phosphodiester bonds, what are glycosidic bonds. So they were not able to understand anything about it. But he was the one, they were the one, this team of Watson and Crick, they really made a big through in the field of biotechnology. So that's the era where the thing starts to go up. Then we never think of back. Things start to develop a lot afterwards. Then in 78, recombinant DNA. That's the also era where the genetic engineering has been started by Stanley Norman Cohen and Harvard W. Bohr. They are still alive. They are American geneticists who develop genetic engineering techniques with the plasmids, cloning, all these. So that's the where the era of genetic engineering has been started from 78. Then site-directed mutagenesis by Michael and Smith. Michael Smith, he's a Canadian biochemist who established site-directed mutagenesis. It means if you're having an amino acid sequence, yeah, on the DNA or the proteins, yeah, let's say you have a big structure of folded structure of protein here, and on that your amino acids are present, like cysteine is there, tryptophan is there, alanine is there, serine is there. So what we do, we make a modification of this cysteine into the serine into the cysteine. We remove this serine and we added this cysteine into that. So we do a site directed mutagenesis. So from one site we remove one amino acid and we added another. Or there are many ways to do it actually. So this is one of the way. So he got the Nobel Prize winner in 1993 in chemistry for the same. Then this famous guy, Carey B. Mullis, he also brought a revolution in the field of biotechnology in the case of producing, the same template of one particular DNA and having a million copies of that, just by increasing and decreasing the temperature, making temperatures to 90 degrees Celsius, then to 70 degrees Celsius, then 50 degrees Celsius, so that it could have denaturation, annealing will happen, then elongation is happen, and then you have millions after 35 cycles or 40 cycles. You have millions copies. You have millions copies out of it, right? And he is a winner of Nobel Prize winner in chemistry for the same in 1993. Good. So that was something, a history about biotechnology, some prominent names who made this field totally revolutionized and made our life easy at the moment. So I always initiate my lectures with these great people with the great stories behind them, because there's a lot of work has been there. It's not that single day your PCR was invented, your genetic engineering, your recombinant DNA was invented, or DNA was invented, or insulin, the sequence, the Sanger sequencing was invented, antibiotics or lock and key model, uh, our Mendelian's laws, or smallpox vaccines. These people have done something remarkable in their life, yes, in order to produce something way out of what we are thinking at the moment. Okay, Anushka Das. I have taken the first vaccine dose. Okay, good. My whole family got vaccinated. I haven't got. I didn't receive the vaccine as I suffered from COVID. And I completely agree with you, sir, that we are guinea pigs for the government. Yes, we are. Nobody is going to challenge this. The hesitancy of scientists to come up with solutions may cost millions lives. I think more than vaccination, awareness is still required. I have seen people wearing cloth masks or single masks still. Yeah, I mean, I was also been part of. I had a little. I didn't had a big thing. I had some kind of low viral fever of that. I didn't have those symptoms like taste going and breath thing or something. But I had a little fever during that time in the last months. So it's a severe thing. Once you had, it's not normal. But what you have to do is during this time, um, just a strong will really helps. A strong willpower. That's the only thing a person could be saved. Um, I was in Kanpur at that time, and I had and that the virus, the second phase that came this time, actually this was spreading in the air, right? It was not like spreading by the human beings by in touch or something. So even though we were wearing masks, we were cleaning our hands, we are not, we can't be saved. Out of four, three people has to get it. I am from Delhi actually. I'm in New Delhi at the moment. So I was going for my cycling in the early morning, 5 a.m. in the morning, and coming by 7. So I used to see bodies, death bodies, every corner. So I have seen the extreme life death rate, unfortunately. I have no words. I don't want to think of that time again. But whosoever has lost their things, I mean, their soul may rest in peace, and their family get enough courage to take that hard time. And those who have survived now, we need to be with good willpower and to start this second life, we can say, because two we have survived two waves of this COVID, the big waves, and we are the one who are survivors. It was also one controversy that it was all done in order to remove people from the earth by giving this virus, so that mainly, yeah, those who doesn't have any diseases like hypertension or diabetes or any other things, they could be removed from the earth. So that's might be that also one controversy is going on. I don't think vaccine is completely tested. I don't want to be lab rat for government. I'm not vaccinated yet. Yeah, it's true. We are not aware what we are getting. But believe me, there's a Pfizer vaccine from USA that has around 91% efficiency. And all the USA states people, they got that one, and it seems to be good so far. And let's see. It is. It's the survival of the fittest, actually. But now it's the thing is the biotechnology, all the related fields of genetic engineering, pharmacology, or immunology, all these fields were not recognized till 2018. There were some forums that were happening, there were some webinars were happening. Very few people tend to join it. Even the government doesn't give funding to it. Nobody gives a damn about who we are as a biotechnology. So I took this field in 2005, and I thought this will be in the future very much essential. But now I can see in 2021, and during the Republic Day, we had one stage show of biotechnology where the vaccines were being produced in that. So it's become one of the essential part of our life, this biotechnology. So it will have a good careers, of course, in future, and just keep continue. So if we see from the perspective of enzyme engineering, um, the definition, the term catalyst, it was given by the Oswald, then lock and key model, Fisher, then Buchner, he demonstrated that enzyme do not require a cell. Then Sumner came, he proved that enzyme is acting like it is actually a protein. Then Koshland, he brought the induced fit model, a much better model than the lock key model, because this was discovered lock and key in 1894. But induced fit, he said it's not like lock and key. The enzyme could change their shape in order to fit the substrate. Then in 63, first amino acid sequence of ribonuclease was reported. Then in 65, allosteric model of enzymes were proposed. Then in 70, immobilized enzymes were discovered. And 80s, protein engineering, chiral compounds, enzymes in organic solvents, polymers. Then in 1990, directed evolution. Then in 2000, computational design of enzymes. Bioinformatics starts to play a role in, because by the 2003 we were like this genome of human body was discovered. Until then, there was very low research publication was happening. Nothing was into the field of research. But since the genome was found out in 2003, your bioinformatics, your molecular biology, your recombinant DNA technology, your diagnosis of various diseases, they just abruptly go up after the 2003. So all these eras, these histories, they really make sense that things develop in really chronological order in order to make things to fit, that each one of us get the benefits out of it. Okay. So who were the main people who got the Nobel Prize winners? Edward Buchner, yeah, Wilhelm Oswald, Emil Fischer, Sumner, Sanger and Tuppy for the beta chain insulin sequence, the Sanger sequencing, Murray and Stein for the amino acid sequence of lysosomes and ribonuclease, Michael Smith for the side-directed mutagenesis, and Carey B. Mullis for the invention of PCR. So these are all people that we have just discussed before, and they all playing a very essential role in the field of our enzyme engineering. So there are two fields, that is enzyme technology versus chemical technologies. So what is the advantages of it? That is, these enzymes are highly selective. Yeah, they can select uh, many substrate. They're environment friendly. They do not produce any waste materials after the reaction is done, and they can catalyze a broad spectrum of reactions. Many reactions could be possible. So less byproducts. It's a non-toxic and non-flammable. Where the disadvantage is that it is expensive, of course, if you want to be environmental friendly, and it's too unstable also, and productivity is quite low also in that case. So some, if there are good points and some sad point too. So this is the nomenclature of the International Union of Biochemistry and Molecular Biology. The EC number, how they give that? The first number of the enzyme, they give the class, which class they belong. This oxireductase, transferase, isomerase. Then the second number, we give the subclass of the type of substrate or the bond that are cleaved. And in third number, subclass of the electron receptor or the type of group are removed. And in the fourth number, serial number of enzymes are found. So according to these four numbers, the nomenclature of enzymes are given. So if we see the basic classification of enzymes, so EC1 oxireductase, EC2 transferase, EC3 hydrolases, EC4 lysases, EC5 isomerases, EC6 ligases. So oxireductase, the name suggests that it might catalyze the oxidation reduction reactions. Transferase, it might transfer some groups. Hydrolase, A's will be always there in the enzymes at the end, because enzymes nomenclature at the end that defines that it belongs to enzyme class. So A's is, it's like my name is Simran. Yeah, if somebody wants to make an enzyme of me, so it will be Simranase. Or for Modi ji is there, so Modi is. I hope BJP government will not pick me up for this. Modi is, because if he's acting like a biological catalyst, it's fastening the reactions. So we call that name to be as with the A's at the end, Modi is or Simranase, anything. Then lysases, which cleaves various bonds by means of other than hydrolysis and oxidations. Then in isomerases, it just changes the structure of that protein, and then ligases, which joins two molecules with the covalent bond. So some examples of industrial enzymes with their production scales. So high fructose corn syrup, HFCS, it's being really highly being produced. You get this into your Bonvita, your Milo that we used to drink in our childhood, which was in the advertisement shown by the many cricketers. That's the source of my energy. Uh, this is all fake. That's all full of sugar, and they have nothing as in known to be healthy for body. So they have this enzyme glucose isomerase. Various companies are being produced. Then lactose free milk. Yeah, who has a lactose intolerance? So for them, this is very essential for them because they cannot digest this lactose in their body because they can't produce lactase enzyme. For them, this is very essential. Acrylamide with the help of nitrulase, cocoa butter, lipase, aspartame, thermolysin, nicotinamide, and so on. So some chemical and enzymatic reactions. Students, we continue further from here onwards in our next sessions. Let's take a 5 minutes quick break. What's the time now? Just get your water bottle or anything. Okay, this is one point. Then we start with the next batch. That is the reactions. So these are the various reactions according to the, if some of you are also from the organic chemistry. I'm not core biochemist. I'm more into the molecular biologist. So my main that is what you say specialty is into the cloning, Western blotting, protein analysis, protein post translational modifications, epigenetics. So I was more into the DNA and protein part. Um, yeah, but I took this subject because I also felt like to have my you know this is also I was needed for my expertise in my research in upcoming time. So I might not be aware of these terms at the moment, to be honest, because what I know, I will say it, which I don't know, I cannot say it. Yeah, so I have to be honest here. So these are some reactions, esterifications, ether cleavage with the EC numbers, and the enzymes that are being produced with them. So this table just giving us a very nice chemical and enzymatic reactions with various examples. So we will discuss now further the background of enzyme engineering, how they are working. So in meantime, just let's take a 5 minutes break here and then we start. So it's let's say 8:10, we meet at 8:15 then. Just quick 5 minutes break, then we start again.
Yeah, students, welcome back.
Oh, hi Shivika Hundo, MSc Biotech from Jamia Millia Aslamia. Good, good, good. You can join me, students, on the LinkedIn to be forever contacts. So I'm sharing some good stuff. Sometimes how to write a good resume. I have recently working on my YouTube channel. Please do subscribe and like these things. It's a lot of effort there actually. Um, this is our live classes going on. So there is a tips for students. So how to go abroad or how to make good sharpshooter resume. So I made some, I'm sharing every day some good stuff for everyone. So just share this link. Please subscribe to the channel, and I will bring some good cover letter also, how to write good cover letter, how to write good, you know. Ooh. Yeah, some good cover letters, good motivation letters for applying abroad or in India, anywhere, for good positions. Okay, let's work on the topic now. The background of enzyme engineering. So it's a selectivity that is the most important when we are seeing an enzyme. That how selective is an enzyme is, and whether it could produce the usefulness of catalysis, the work that we want out of it, whether it will work in that way. So all the organic chemists, they give highly the energy towards the catalyst productivity, its activity center, and it could be recycled, how much it could be recycled again and again. So these are the key parameters for its applications. So these terms were given by Jepson and Beller, and was given the statement about the productivity and biocatalysis of the enzyme in 2004. And this is the industrial production of hydrolysis enzyme, industrial biocatalysis in the new plant in Geismar, USA, which has a capacity of production of 2.500 trillion per acre. And this is, they are producing here herbicide in these big containers. So it's a big field. And other way around within these in the case of production, so carbohydrates play a major role. Then comes the amino acids in the production. Then comes your secondary alcohols, peptides, beta galactans. Then further chiral compounds, other non-chiral compounds will come. Then in the less ones are steroids, fat derivatives, and nucleotides. And from the biocatalysis production, the main markers that are being produced are from the pharma industry. Then comes other factors like agriculture, food, cosmetics, polymers, feed, and several other sectors. But majorly are the food, pharma, and agro. So if we see from the biotransformation perspective, your hydrolysis enzyme, it's brought to the largest concern related to this, around 65 or 45 percentage. And then there comes other one, oxidizing cells and reducing cells, which brings about 28%. Then isomerase is around 4%, and lysis is 5%, transferase is around 11%, and oxidoreductase even less than that. So in this way, our whole classifications of enzymes, whole enzymes being that are classified, they are moved into that field. And there are two types of autotrophs within this field of natural, biotechnology. That is chemoautotroph and photoautotroph. The chemoautotrophs, they are the one which takes energy from the chemicals to produce the food, and the photoautotroph, they took the UV light or sunlight in order to produce the energy. Yeah. So in chemoautotroph, what is happening is they're taking carbon dioxide from outside, yeah, and hydrogen from the outside, and they're producing ATP with the various reactions of thiosulfate chemicals also, CH2O, and the ATP is being produced inside. And also then H2O is being lignin monomer organic molecules are being inside, which are doing these reactions. Then in the case of photoautotrophic, your light is being coming from outside, and then with the help of your Golgi body and nucleus, your ATPs are produced. So that's the main difference between both of them. So type of reactors that we use in the industrial biotransformation, this is from the fermentation perspective. The highly used fermenters that are used in the enzyme engineering are fed batch reactors, then the fed batch, then continuous plug flow, continuous stir tanks, and some are still unknown. So majorly batch reactors are being highly used in this field. Then types of biocatalysts that we use in the industrial biotransformation, that is free cells. Mainly the free cells are the highly productions in many processes they are being used. Then comes your free enzymes, immobilized enzymes, then immobilized cells afterwards. So in this way they are being divided. Then enzyme activity is depending upon many factors. That its activity could go up and down based on the type of fraction they are going, type of mechanism, kinetics, dynamics as it is. Whether it's a glycosylation is happening, phosphorylation is happening, dephosphorylation is happening, what kind of pH is there, whether it's a redox reaction or is this an oxidative reaction, what is the expression of your cells are. So all these factors, they really matters to your enzyme activity. And these are the typical parameters from the kinetic perspective they have told. Michelson constant, Michelson-Menten constants, Km is there. Catalytic equilibrium constants, Kcat is there. Stability is there. The substrate and product production is there. So according to these matters, your enzyme activity depends upon. So what productivities need for synthetic application? So this is from the industrial perspective, that how much they are being produced, like acrylamide, acetate, citric acid, riboflavin, into the industrial processes, how much they are being produced. So far, phenyl ethylamine is being highly produced from the biotechnology perspective, and from the chemical perspective, acryl, nitrile, methanol, and ammonia, they are being highly produced. Ammonia is the highest production so far. And on the other hand, the volumetric end from the annual production, amino acids, they are producing around 54.6 gram per liter per hour. They are the largest production of the in the field of enzymes. So like decarboxylase, oxireductase, amidase, lysase. So all these enzymes are highly produced, and these data are not from the India actually. These data from the USA. So these presentations, these from papers made from the recent USA data. So in the USA, these are being highly used at the moment. Then comes your alcohol, carbohydrates, nucleotides, and so on. So that's the something a little bit like in touch with the figures and numbers. How things are being produced with the respect of fermenters, which fermenters are being used, which kind of your enzymes are highly used in the industry, what kind of products are being produced, which industry are being affected with the enzymes. So we just had touch a little bit about enzyme engineering perspective from the industrial perspective, how industry really works within it, how much volume really matters, how much yield really matters in the industrial perspective, from the industrial perspective. Then fundamentals of protein chemistry. So we know that there are around 20 amino acids with non-polar and polar amino acids. These non-polars are with the aliphatic alkyl group. So here we can see this glycine, H, alanine, CH3. There is one amino group, carboxyl group, and H. And according to this group, R group is being changed. You will have various kinds of amino acids. Polar, non-polar, positively charged, negatively charged, aromatic, and so on. So that's it from the history perspective. Some basics of industrial enzyme engineering. Little bit, because these amino acids, they are very essential uh, from the industrial production of enzymes, because of their alkyl group, because these alkyl groups give them different properties, different charge, and different output. So together these negatively charged groups, they are aspartate and glutamate, so they together will have their own functions. On the side, positively charged are groups like lysine, arginine, histidine. So they come into this group. Then is aromatic group, which are having these aromatic rings in that 6 chain rings, and here 5, and 1, 2, 3, 4, 5 ring here also, and this is 6 ring. So yeah. So we'll share this now. Yeah. So student, if sometimes if I'm not there in the class or something got mishappen, just message me or call me, because at the moment in the 3 days I'm having many workshops: enzyme engineering, molecular biology, and also one more medicinal biochemistry, and also having 1 month course of biomedicinal chemistry, and also these workshops from the uh, MBBT June batch, AIMS MBBT June batch. So I'm going through many classes at the moment. If something goes up and down, so please call me or let me know if I'm doing something wrong. So my 3 days gonna be a lot of many work down there. Okay, cool. So the basics of history of enzyme engineering, of enzyme history, and its basics from the industrial perspective. We are cool with that now. So I will take you to the next lectures of what do you say, mechanism, how the mechanism of enzymes are working. That's very basics that I will take you now. Actually, not so something in advanced version. So my reason to take these classes here was basically to take you to the to clear the basics about enzyme engineering, like how things are working. These enzymology books, yeah. So so history is over. Now comes the mechanism. So in this part, we will discuss uh, mainly about various apoenzyme, holoenzyme, prosthetic groups, enzyme specificity, how the activation energy is working. We will discuss about the structure of enzymes, mechanism of action, and we will discuss the classifications of these enzymes too. So the outcome would be we will be able to understand the energy of activation, what are the structure of enzymes look like, how the mechanism is working, and get to know about a little bit about classifications of enzymes. This is from our previous universities. I will remove these codes later on so that you can have it later. So enzymes, so as we have initiated already in the morning, that they are playing a key role, important role in our life, whether it's a metabolism, diagnosis, or therapeutics. This we have this KFT, LFT test. Whenever you go into your diagnostic lab and the doctor tells to have these multiple tests, before going for those multiple tests, just ask the doctor itself whether they make sense to do all the tests just in one go. because some tests are done in order to fulfill the doctor's fees, because doctors have very good connection with the diagnostic labs. The more test he writes, the more get money he will make out of it. So make sure the test that he's writing to you, you're all biotechnologists, you are yourself are now upcoming, yeah, you know many things about medicines now. So make sure that the test that you're going, they are the good one that you want to go for. And you can also diagnose yourself by reading these tests, by reading the values, whether it's high or low. If it is high, what's the symptoms of that? If it is low, what's the symptoms of that? So yeah, at least all the biotechnologists, all the geneticists, all the students who are studying these, they could read their families' medical, conditions, how they are, and good for us. So in that sense, enzymes, I want to say that they are playing very essential role in many fields, and they catalyzed many reactions, biochemical reactions in the living organisms. And the level of enzymes in blood are of diagnostic importance. Example, it is a good indicator in disease such as a myocardial infarctions. So during the heart attack, the level of enzymes, your phosphatases, your kinases, they go up and down. They go really up and down. If somebody gonna get a heart attack, so the level of phosphorylation will be very low. So in that sense, your phosphatase enzymes will be very low. So so we get to know about many things. Just a second, students. Hello. Uh, Good morning, sir. बढ़िया सर, बढ़िया सर। हाँ जी, बोलिए सर। हम्म। जी जी। हम्म। ठीक है। हाँ हाँ, मैं कर लेता। हाँ सर, आपको 9 9 सवा 9 बजे कॉल करता हूँ, अभी क्लास चल रही है मेरी, 7 to 9 वाली। तो उसके बाद आपको मैं कॉल करता हूँ अभी। ओके। So yeah, and also therapeutically in the digestive enzymes, they are playing an essential role. So if enzyme is there in our life, then we have life. If they are not present in our body, then there's no life. That's I always say. Enzyme yes, life yes. Enzyme no, life no. So they are acting as a biological catalyst. They come into the reaction and they do their work and they get out of it. And what they do is they make the rate of reaction by lowering the energy of activation. They make the reaction of your protein reaction, it increases it up by making the initial boost that is required, lowering it up. So how from the daily life, let's say an example, one person wants to carry a big luggage over the first floor. So that person is doing that work on his own, yeah. But what he gets a pulley and he goes to the first floor, and with that pulley he like make a knot to the luggage and get it up very easily. So to initiate the reaction that is happening, to get the luggage up to the first floor, the reaction got really fast, yeah. And the effort that he has to put initially, it also got lower. The energy of activation in order to make the luggage come up, it's work. So then pulley in this case acts like a enzyme, and your luggage is your substrate, which is coming and working, it's playing as a role. So in that sense, your enzymes are working. So they catalyze nearly all chemical reactions in our body, and they do not alter or consume during the reaction, so we can use them multiple times and they are reusable. And these enzymes, they had really important place called as active site. Yeah, they have this active sites. That's the area on the enzyme where the substrate attached to it and is called as the active site. So enzymes are usually very large proteins, and the active site is just a small region of enzyme molecules. So the area on them where the substrates attached is called the active sites, and they are usually very large proteins, and active site is just small region of the enzyme molecules. So here's the active sites we can see in the reactions of enzymatic reactions. So enzyme molecules, they contain a special pocket or cleft called the active site. So this is the active site here, and this is our substrates. So we can see the shape of the enzyme is not so like in the same shape. Yeah, it's somehow to that shape, but not fully to that shape. So here what we will do, substrate will enter to the active site. It will make the enzyme's shape adjustable to its. Once it is done, once the product is produced, your products will get out of the enzyme. In between, they will have this transition state. This transition state is called as enzyme substrate complex, and this transition state is called as enzyme product complex, right? So these two enzyme and they are very unstable states. They are not prominently happening in the in the reactions. On the other side, this is the um, yeah, empty one. So in this way, basically your whole enzymology or whole enzyme engineering is just based on this principle. Your Michaelis-Menten equations, your competitive inhibitors, your allosteric enzymes, um, your Lineweaver work plot, all the study that we will do in our upcoming lectures, they are all depending upon this principle, that substrate came to the active site, it's get attached to it, and then it makes product, then become enzyme product complex, and the products are relieved, and this enzyme could be used again for the same reaction, and it will be used multiple times, like millions times maybe within hour, and yeah, and lets you have products in a huge amount. So what is happening? The same thing is repeated in a different way. The substrates get joined to the enzyme, and the reaction starts to work on. Then comes your lock and key model. In this lock and key model, basically the active site has a rigid shape. So that's the lock, key, and model. They say that that they have a very specific shape. It cannot be changed. Yeah, it has a rigid shape, and only substrate with matching shape can fit. So only the one which match it fits, only those will fit. And the substrate is a key here, yeah, and the log is the active site. So this key is going into that, and it is being fixed because of this shape, correctly fit. But this one, it doesn't have this thing. This is missing, so it's not there, so there will be no reaction. It's also another example is like gloves that you wear in the lab or during the winters. So our hands goes into the gloves, and after some time, if they are tight or they are loosen up, so our hands take some time, to adjust to that. But these are not rigid. So that's the example of gloves will be your induced fit model that we will discuss in our upcoming presentations. Then, because this was discovered in 18, like we have discussed in our first lecture, in 18th century somewhere, but then came that no, these enzymes are not rigid. Actually, they could be fit. If we add our enzyme, they could be some changes could happen at the structural level, and only then enzyme could attach to it and makes the reaction done. So this explains in this lock and key model that enzyme specificity, enzymes are very specific, and this explains the loss of activity when enzyme denatures. When enzyme is not working anymore, there is a denaturation is happening, so reactions will not take place. Yeah. Then comes, this is very apoenzyme and holoenzyme. So the apoenzymes, so what is the difference between them is, the enzyme without its non-protein moiety is termed as apoenzyme in active states. So this is your apoenzyme, right? which is of a protein portion and is inactive, right? This is an apoenzyme, which is a protein portion which is an inactive, and this is coenzyme, which is a cofactor, which is a non-protein portion and which is an activator. So this substrate, it comes on top of it, and the whole enzyme will be active. So this coenzyme, it goes into this, which is a non-protein part and which is an activator, and then your holoenzyme, this whole reaction of this active site will be worked out. Then comes your cofactor. What are cofactors are? They are the non-protein chemicals that is bound to the enzyme, and it's of two type, that is your coenzyme and prosthetic group. Coenzymes are the one which are loosely bound to your apoenzyme by the non-covalent bond, where the prosthetic groups are the tightly bound to the apoenzyme by the covalent bonds, and they are called as prosthetic groups. So example of coenzymes are vitamins, compounds derived from vitamins also, and prosthetic groups are like manganese, copper, FMN groups. So all these belong to the prosthetic groups. Now comes your enzyme specificity. So enzymes have varying degree of specificities for substrates. So they could recognize and catalyze the reactions depending upon how many substrates are there, how many similar substrates are present, and type of bonds they are making. So it could be enzyme specificity could be three types. There is absolute, group, and linkage. Beyond that, yeah, absolute, group, and linkage. And in absolute, this catalyze the one type of reaction for single substrate. And in the group one, for the group of similar substrates, it's catalyze the reaction, and the linkage for specific type of bonds. So as we have discussed in previous thing, that enzyme could recognize based on the single substrate, group of single substrate, and particular type of bond. So based on that, we have divided into them absolute, group, and linkage. And in this one, we have single substrate, we have similar substrate, and we have a specific type of bond in it. So in absolute, you have example called urease, which catalyze only the hydrolysis of urea, and in the group, it's a hexokinase, which adds a phosphate group to the hexoses. Then comes the chymotrypsin, which catalyzes the hydrolysis of the peptide bonds. So important terms to understand the biochemical nature and activity of enzyme reactions. So activation energy, what is it? It requires some energy to get them started. Like anything, like we have discussed the example of a luggage needs to go from ground floor to the first floor. So the energy that requires, it could be manual or it could be done with the help of pulley. So we can use that enzyme or we can use it without enzyme. So there's uncatalyzed reaction and there's catalyzed reaction. So the first push that is required to make your reaction done, this energy is called as your activation energy. And then what we have just discussed so far, that the enzymes are of two types, that is complex or holoenzymes, and then there is a simple only proteins. The complex or holoenzymes, they are the protein part, and they are non-protein part called as cofactors. So they are apoenzymes and cofactor. Within the cofactor, it's of two type, that is prosthetic group and coenzyme. And the coenzyme are the large organic molecules which are loosely bound to the apoenzyme, whereas the prosthetic groups are usually the small inorganic molecules and they are tightly bound to the apoenzymes. Yeah, that's the summary of what we have just discussed of all the apoenzyme and holoenzyme there. Cool. Okay. So we have just discussed that and the enzymes, they helps to increase the rate of reaction by decreasing the activation energy. That's the main soul of this enzyme activity. Yeah, whether you open any book, like I have this book called Lehinga, maybe it will be helpful for you. I don't know, can we share big books in it in the group? Okay, within this book, if you go to the chapter 7, there is a chapter 7 or chapter 6, sorry, enzymes. So I am just discussing everything from this chapter only. Introduction to enzyme, how enzyme works, enzyme kinetics, example of enzymatic reactions, regulatory enzymes. So this will give you a brief, just in your free time, you can study about all these things from this chapters also. So I'm just going through the Lahinga chapter. Um, how the enzyme activity is working from the enzyme perspective. So I shared this book already with you in the group. It's a chapter number 6. Then we have just discussed that there's a lock and key model and induced fit model. We will discuss this induced fit model in upcoming lectures. So how does this enzyme lower the activation energy? That's the question. So let's say an example, there's a man who is having this boulder over the hill, yeah, and he wanted to push it up and make it to the top, and then afterwards it could go slide down. So he need to push some energy, he need to put some energy to take it to the top. Yeah, it's a lot of energy is required to make it to the top here, and yeah, and make it here. So it needs lots of energy there. So this is the time, this is the place, this much energy from here to there. This is the activation energy without enzyme. So we didn't add any enzyme in this. So this much energy is required to go up. And this one at the down is the net energy that is released after splitting of these enzymes. So here's on the top is example, your lactose, which is a mixture of glucose and galactose. It's got break up, the bonds got break up in order to produce glucose and galactose. It will produce glucose and galactose. So this is done in the absence of enzyme. So in energy that is dissipated in after the splitting of lactose, so that much of energy is released later. So but when we have the enzyme called lactase enzyme, it will attach to this substrate of lactose will attach to the enzyme like this, and it will produce glucose and galactose, and you can see the amount of activation energy when the enzyme is present is abruptly decreased. It's abruptly decreased, so less, yeah, and the net energy that is released is very less as compared to the previous one. So the energy that is required now is just he doesn't need, he can even push it with his legs and things could go. So that's the functions of, with the help of enzyme, you can make your reaction done. So maybe this is the last topic, we will discuss it today, and then we do it the rest things tomorrow. The same thing that we have just discussed, it's in the form of green energy, free energy, there's a delta G. Yeah. So, and progress of reaction. So in the case of without enzyme, you can see that without enzyme you need higher amount of activation energy, and then it goes up down. But in the case of enzyme is present, there's a less amount of activation energy. So like this in this case, and in that case it's even higher. So this is the activation energy without enzyme, this much, and this is the activation energy which is now much lower in the presence of enzyme. Whereas the down energy that we are discussing, the net energy released afterwards, that is known as delta G, is not affected by the enzyme. So this will remain the same. So that's the basic idea behind the activation energy. So we discuss the rest part later in our lectures. Yeah, so I will share these presentations with you soon. We'll just remove the logos of that and then we'll share it. And then, okay class, that's it for today. I hope you had a quite good basics understanding of enzyme activity, how they are working. Maybe you rebrushed your previous lectures, and eventually we will discuss about kinetics, purification, isolations of proteins, some more yeah, like chromatography and all these techniques in our upcoming days. Cool, then if you have any questions, we are open for the questions in the last 5 to 10 minutes. I can take your questions now. Okay, if no, then take care. Then we see us tomorrow at 7 a.m. in the morning with the fresh startup again, and then we continue with that. Thank you. Take care. Bye bye. See you then. Wish you a nice day ahead. Stay safe.